So I begin with thanks to my collaborators, David Steinsaltz and Steve Evans, who don't see eye to eye with me on some things and that I'll talk today. And thanks to Ian Matheson here at the Simons Institute, who's been explaining a lot of things to me, to my student Emil Hirati, and to Richard Sussman, who is the patron of all this work that brings biodemography and genetics together. So today is a little different talk from many. This is very much work in progress or even thoughts in progress. There is a paper that will be coming out in PNAS, but this is from the speculative part of the end of the paper in the discussion. I'm talking about the same model that David talked about yesterday at the end of the session. And you might have been left with the impression that the model doesn't work, that there are all kinds of pathologies. Well, the pathologies are what are mathematically interesting, but they can be avoided in perfectly straightforward, sensible ways by allowing for small deleterious effects at younger ages whenever alleles have effects mainly concentrated at older ages. So if you remove the unrealistic feature of a sharp cutoff of nothing up to some age and something beyond it, you get rid of that unrealistic feature, then the models perform very well. They generate very good-looking adult mortality schedules. One can tune them to fit schedules, both for humans and for many other species. I use them in classes constantly. They easily predict a wide range of empirical behavior without pathology. They behave beautifully 99.7% of the time. But the 0.003% of the time is what's mathematically interesting, that if you make the assumptions David talked about that mimic the assumptions that used to be made, then the models break down. So we know we've got something more than we used to have. But what we mostly have is a flexible way for incorporating age specificity into the story of mutation accumulation. And now, although the model applies to deleterious alleles affecting fertility and juvenile mortality, I'm going to talk about a special subset of those alleles and concentrate on adult mortality today. And so, David talked about the mathematics, and I'm going to assume that whatever mathematics you wanted to hear, you gained from David, who really invented the model, and with a lot of help from Steve and a little help from me, proved the main theorems in the model. So you've heard that, and if you still want more, there is a memoir for the mathematics. But I'm going to talk about empirical tie-ins. And although I've said the model is flexible, as David emphasized, it depends on having some specification for the age-specific effects of deleterious alleles. And these are small effects which we can't detect, and so there's no empirical, simple empirical way of finding out what those shapes should be. And so to ground the model in empirical reality, we have to look toward macro predictions, toward a broader brush piece to see what has to be true for the model to be compatible with what we can see, especially from genome sequences. So that's the problem I'm talking about today. I emphasize we have a model that works well for demographers, is now in broad use, at least for people who come from Berkeley. And now we're trying to tie this theoretical work to observation. And so I want to talk about, and that's not easy, I want to talk about the challenges. And so really I'm looking at 4 questions to which I will propose tentative answers, which you are free or encouraged to shoot down or revise. So one's about the flow. So I'm looking at a special class of deleterious mutations, these MA alleles that I'll talk about a little later. How many of these per individual, per generation are coming in? Overall mutation rates are thought to be something like 70 per person per generation now. How many MA, most of them neutral, how many MA alleles. Stock, how many are we carrying? How many do you have inside you? Do I have inside me on average? Selection, how big is the range of selective costs that will fit into the model? And along with that, antiquity, what is a typical age for these alleles? The most pressing issue for demographers, because it's perfectly clear that humanity, at least, is out of mutation selection balance now, that the onset of agriculture and civilization have enormously changed the selective pressures on any of these physiological pathways. So the antiquity of the alleles is the key to their demographic relevance. Now, I'd like to preface my talks by pointing out that Darwinian's natural selection is a story about demographic success. It's about the survival and offspring that one has, that organisms have across the life course. And these age-specific schedules, as David encouraged us to think yesterday, are the proximate determinants of fitness. That's the offspring carry the genes onward to the next generation, are born at ages of their parents. It's an age structure. And so natural selection in the first place is operating on and reshaping age-specific demographic schedules. And the whole point of what makes this interesting is that that age specificity, as David explained, means that there's always epistasis. Anything that is having age-specific effects on fertility and survival is going to affect alleles that are having effects on other ages. So, in honor of my PhD dissertation supervisor, John Kingman, I wanted to smuggle in a coalescent tree somewhere, and this is a coalescent tree for most recent common intellectual ancestors. That is, the MRCIA. And so, on this right here, the leaves are demographers blowing around across the parking lot, and on the left here you see the leaves, a lot more of them, that are geneticists. Lots in this room, this side of the parking lot. And let's say I'm here, and so a coalescent tree heading back to Sir Ronald Fisher, who cared a lot about age-specific schedules, and indeed his age-specific reproductive value function has become a cornerstone of demographers' stable population theory. And Fisher also did some genetics, and so he becomes an MRCIA for all of us. But this tree suffers from incomplete linkage sorting because there has been some lateral gene transfer very recently from Sir Peter Medawar, who developed mutation accumulation, and J.B.S. Haldane, who worked out relationships between rates of new mutations and total loss in fitness from deleterious alleles. So that's where we come together in this subject. We're going to have the legacy of Fisher with lateral transfer from Haldane and Medawar. Epistasis, David explained it in much more detail, and remind you, we're looking at a framework that became, I think, current from Kimura and Maruyama, but to which we have added rapid recombination. And so we're looking at weakly selected, semi-dominant deleterious mutant alleles, random mating, diploid inheritance, but the selective function is working on haplotypes. And so in that setting, there is the classic expression for non-epistatic multiple alleles that in every textbook, where the net reproduction ratio, the measure of fitness, is just the product of these individual costs, and that can be approximated for small s with an exponential function. And in our setting, we have a fitness specification that's strongly analogous, but instead of just having the exponential, we have an integral outside the exponential. These are profiles that act. M is indexing, well, I'll talk about what M is indexing. We're summing over alleles and integrating it against a baseline. And so this piece is strongly analogous to what you've all seen many times before, but it's the averaging over a life course that generates epistasis. And I should mention that the model is in continuous time. I'm trying to draw lessons about discrete generation outcomes, and this, as my colleagues will tell you, is not without challenges. Okay.
If we're going to comparing to like empirical data, is it obvious why we can ignore dominance?
No.
Okay.
That's a complication that, that waits for later investigation. We've got enough problems before we go there. I don't think that's a difficult problem. But basically, we're going to do selection on haplotypes, not on the diploid genomes. So, it's a story about genetic load, but there are four special features. There's a lot of literature coming out about genetic load and its relevance right now, and we've benefited from a paper by Agrawal and Whitlock, a review article that many of you have seen. But our piece fits into a special part of that study of genetic load. David described our objects as mutations, but I've come more clearly to think of them as teams of mutant alleles. What is being indexed here is a team of mutant alleles drawn from widely separated sites, and what the members of a team share is their age-specific profile. So we have lots and lots and lots of sites, and then we pick out those scattered sites whose age-specific effects are all similar. So we cluster sites by their age-specific outcomes, not their physiological pathways. They're operating through very different physiology. No physiology in common, perhaps, but outcomes, age-specific outcomes. We cluster those together, and then for each team of alleles, we have some count. And we think of a lot of sites going into the count for one team. And now I'm restricting attention to a subset of the load, which I'll call MA alleles for mutation accumulation. And these are the ones that are mildly deleterious, held in mutation selection balance, and affecting adult mortality. So juvenile mortality and fertility are another part of the model. We're starting with adult mortality, which, as David pointed out, has great regularity among human populations and similarities with a number of model organisms. So we're looking at the part that is most similar, part of the mortality schedule, part of the demographic rate schedule that is most similar across species. That's our starting point. And the advantage, this is going to have some advantages that the people thinking about genetic load don't usually have. They have a problem with what on earth could be a load-free genotype. But for adult mortality, we're not going to have that problem in quite the same way. And that comes from the fact that the highest survival probability for an adult is 100%. You can't have more than 100% survival. So there's a bound on what the load-free genotype can look like. And then the second advantage we have is that for our epistatic setup with the formulas that David gave you and I recalled for you, we have a closed form expression for the adjustment that you need to make in Haldane's principle for epistasis. So instead of talking about the modifications due to epistasis in a general way, we can actually calculate it out. It's a clean, closed theorem. And now also coming from demography across the parking lot, we just assume that prehistoric population growth was kept near zero. So you have density-dependent feedback, size-dependent feedbacks, other kinds of feedback systems that over the long course of prehistory let fertility and juvenile mortality adjust to resources and predator threats. And so we're not looking at loss of viability from the accumulation of mutations. Populations compensate for that and keep the growth rate zero. There's enough potential that it's resource base and the environment that governs the overall growth rates. So there's Sir Peter Medawar, just to have someone on the have a picture somewhere in the slides. And this is the graph I mostly want to talk about. So remember, we're going to have an attempt to come up with rough guesses at flows, rough guesses at stocks, use them to make rough guesses as where in the range of selective costs are the alleles I'm talking about, and end up trying to guess what the antiquity of many of those alleles might be. That's our four-fold mission here, and the first mission is flows. What's the flow of alleles? So there are lots of mutations, mostly neutral, many entirely neutral, coming in that are measured in contemporary ways, especially from actually linking families and identifying new mutations, 70 or so per generation per person. But I'm going to argue that my MA alleles are a very tiny piece of this. And I think we have a way of getting at the biggest share they could be per individual, per generation on this. And here's the idea. Anthropologists, bless their hearts and souls, before existing surviving hunter-gatherer tribes were changed by too much contact in the early stages of contact, did a lot of measurement of life tables and fertility schedules. And so we have a number of empirical life tables for hunter-gatherers, contemporary, not prehistoric, but they give us some guide to hunter-gatherer lifespans. And here is the average survival curve, probability of surviving, that was estimated by Michael Gervin and Helly Kaplan from 5 fairly carefully measured anthropological hunter-gatherer schedules. I'm not talking about juvenile things, but this schedule is essentially driven by a Gompertzian adult mortality curve. That's what they estimate. And so plus a background extrinsic mortality. So a constant level plus an exponential. And now let us suppose that at least some piece of that background extrinsic mortality has nothing to do with genetics. Let's say at least half of it doesn't. It's the tigers out there, or mostly not tigers, but alligators in some of these populations. And if they happen on you, they get you. Well, that then gives a kind of maximal level that if for whatever the genetics is doing to reduce mortality at older ages, the most it can do is take you down from what you'd have anyway. Could be 100%, that's not from here too much, but I'm going to let it go down a little to get to what you observe. So that's the most that these alleles can be doing. And probably they're doing less, because probably the resource allocations and a number of other adaptive processes also imply, as David mentioned, also imply that you're going to have some rise in senescent mortality, rise in mortality with age. It's not just genetics that's doing this. So probably what we think of a baseline without these alleles should be somewhere in the middle here, but the most you can get is that. And that's going to play off against a fertility schedule. But when you talk to anthropologists, you don't just want offspring, you want nurture. So anthropologists love to study parents and grandparents, the grandparenting hypothesis in social species. So I have taken the demographer's a standard Cole-Trussell natural fertility schedule without contraception. And then I've put in some smooth allowances of a sort for the good effects on your descendants of having their parents and their grandparents survive. So, somewhat speculative, but this is in the range of shapes that everyone thinks about. And then, what do? Well, we can compare. First of all, we can compare the net reproduction ratios if you had this mortality curve compared to what you can have with this. That gives you total fitness cost. And then, and in this example, that comes out to be about 0.13, about 13% loss of net reproduction from these adult mortality things. If you push the worst of the mortality that was observed in these 5 populations, you can push that up to 0.3 or something, but you can't push it too much higher. And then we have a closed form formula, which actually looks a little like an entropy. I won't go into it, but that's what equates the total rate of mutations into teams of mutations, not to the total loss in fitness, but to a transformed version of that. And that gives you about a factor of 50% more. So epistasis is giving you about 50% more. You need a higher mutation rate because the more mutations you have, the less effect each one of them has. And that gets you in the range of 1/5. 1/5 of one of my MA mutations per time. And if you push it, you can get it up to 1/2, but I don't see any way to get it much above 1/2, not 1, not 2. Out of those 70 things, we're looking at per generation, we're looking at a small portion, maybe one every 5 generations coming in for this subclass of mutational load. Now that's important, I think. I hope the people who work on genetic load listen to us, because they are worried about the problem that so many mutations are coming in that it doesn't seem at mutation selection balance that we can get rid of them. We have to knock off 80% of the possible offspring all the time. But for adult mortality, we don't have this problem. 1 every 5 generations is not such a wild thought for a genetic cost on adult mortality. That's something you're carrying that's not quite right. 1 in 5 generations will kill you off in an age range that matters for your descendants. It seems to me that's a number that doesn't cause worry in a reasonable ballpark. Now that was flows, now we need stocks. So this sets me trying to understand genetic literature. And again, many thanks to Ian, who's been helping me with this. So they're now whole genome or exome sequencing studies, and so people are actually classifying alleles, single nucleotide variants, and work by Tennyson and another by Nelson in this issue of Science. This is the one that I started drawing on, and it's the only one I know that presented a published estimate of the numbers that I wanted. So in background there, it's only the exome. They have 2,044 people split between European and African descent. Found a pool of 500,000 single nucleotide variants that you could find with this sample size. On average, 14,000 per person, mostly neutral. This is my other picture here. This is going to be a distribution by selective cost times 10 to the minus 4, and a lot of these are down by 10 to the minus 4. But Tennyson and company is 22 of his friends, gave numbers of between 300 and 600 for average numbers of functionally deleterious SNVs, which were carried by individuals on different criteria for what one counts as functional. So hundreds, not thousands, not dozens. Now, these aren't quite the numbers we want, but they're closer than anything else I've found. This is just the exome. And we're talking about processes that probably have a lot to do with gene expression and regulation. So we'd expect there to be a lot of mutant cases outside the exome, but maybe not huge factors. Maybe there's 1,000 of these total, something like that, rather than 300. But then we're restricting to MA alleles, so that brings us back down again. Again, we don't know how much, but perhaps a third, a fifth, maybe more, maybe less, but not perhaps a tenth or 100th. I think we're better than within an order of magnitude. For the astronomers among you, I would say we're in an E-folding, a factor of E for these numbers, perhaps. And so if you put these together, we have 0.20 coming in, and we have something like 300 hanging around. So a selective cost is coming out to be something like 10, some multiple of 10 to the minus 3. Well, a multiple of 10 to the minus 3 is what makes demographic sense. But when I did these calculations, I didn't understand that that's what I wanted it to come out at, I promise you. It came out, and then Ian explained to me that that is what makes sense. So in these neutral, so we think of, for European populations, or we think of effective population sizes of 10,000, so we're in 10 to the 4th. At 10 to the 4th, drift is taking over. So when you're down here, things at the 10 to the minus 4 level, that's effectively neutral. Over here, up here, when you get to 10 to the minus 2 level, those are going out in historical numbers of generations. That's strongly selected things. That's things you can see in GWAS studies. That's over here. And our pieces are coming in 10 to the minus 3-ish in the middle of the board, where we want them to be, where I'm talking to you as. So I found great encouragement from that notion that we seem now to be talking about a regime where what matches what one's seen in the sequencing studies is compatible with what the demographic story really needs. We're 10 to the minus 4, a model that has no drift would be useless. If it were, that's over here, if it were 10 to the minus 2, we'd be talking about very detailed physiology. Instead, we're talking about a statistical accumulation of small things. And so, in the general way, the antiquity of alleles is under selection, and right, Fisher models with selection is somewhat proportional to the inverse of the selective cost, and we have an actual, in our equations, that comes out to be true, average selective cost. And so in the time scale then, 1,500 generations, some of them, there's a huge variation around them, but a lot of this is an ancient legacy operating when mortality was severe enough to be like hunter-gatherer levels, that you lost people during reproduction. And so these proportions were putatively shaped in these high mortality regimes before all the changes that you see from civilization.
Why are you ignoring the change in population size?
I'm not. You have set me up for it. So now comes the point. This goes on up to here, and what the writers, Tennyson in particular, say is explosive population growth. This is a fraction of 0.001. Right? 1 per 1,000 population growth is explosive in this world, and not in the world that contemporary demographers work with. So when they found this, they hypothesized a very large number of these SNVs are the result of this signature of explosive growth. And then there have been a series of papers looking at this. One of the ones that I've studied most is by Gatzave and is it Andy Clark and people, but there's another by a person named Simons, Yuval Simons. These may all be people known to you, but they're new to me. And what they mostly find is that the large increase in numbers of single nucleotide variants happens largely down here in the nearly neutral range. So there's a huge increase, can be attributed, this is simulation work on models, can be attributed to changes, recent changes, being last 400 generation changes in population size, and that the numbers here in the middle of mildly deleterious alleles are much less strongly affected. So that's what I read the literature, this is still coming out, that's what I read the literature to say, that the recent population growth is affecting mainly the ones down here. Not a closed issue, but that's what I read the new studies to say. So coming back and summarizing, what looks to me to be the case, how many per individual, something like 1 per 5 generations, how many of these particular adult mortality affecting alleles, something in the hundreds, selective costs well into the mildly deleterious range that you won't see in GWAS, but which will contribute to heritability and will presumably not be tied to very special big things that depend on today's environment, like the presence of chocolate bars or something. Right? Things that have big effects today are not in this picture, and enough of them well back in prehistory that you have a historical story.
Sorry, can you say something about how you're calculating the antiquity of these things?
How I'm counting?
Calculating the age of these mutations.
Well, I'm not going to say a lot. This is early modeling, but the basic picture is 1 over the average selective cost is going to be an average age for these mildly selected pieces.
But you need to take into account the population growth in that sort of thing. Is that
Well, if Gatzave and Clark and Simons and others were finding that the last 400 generations mattered, I would need to do this. If it doesn't matter much to this regime, which is not my claim, it's just my reading of the literature, then the reconstruction of these populations, and there's a long paper by Clark and his team, reconstruct the European-based population at pretty close to, with a bottleneck, and then pretty close to an effective population size of 10,000 for most of that time period. So if I get back behind 400 generations, I'm understanding that I can pretty much let ENSA be not over here, not for the nearly neutral, but up here for those things that are well better than drift, that I can as a first cut, just take 1 over S. Now, Monte Slatkin has been here and others. I'm aware there's a literature on estimating allele ages, and there will be, from the site frequency spectra of these, one should be able to do much better, but in first approximation, that's what I'm taking.
It's tough. I mean, they're not so far apart, right? 400 generations and 1,000 generations.
Okay, I mean, I welcome discussion of this. I'm just saying where the point at which I've arrived. For demographers, I should say, these growth rates less than 1 per 1,000, half per 1,000, quarter per 1,000 are pretty small. But it's clearly a question, how much do they one could do some, I've done some simulations myself, and they do fancy simulations and see how much difference it does make. So I'm just ending up with a piece, the frontispiece from the book that incorporated demographers into this world, a book called, a collection called Between Zeus and the Salmon. Zeus, the exemplar of a creature who lives forever, and the salmon, the exemplar of a creature who spawns and dies. And for demographers, the goal has been to find our place between Zeus and the salmon. I'm encouraged that genome sequencing studies tied in with the mathematical models that, in Fisher's tradition, recognize the importance of age, seem to me to be giving us a reasonably consistent picture of the genetic, likely scope of genetic influences on adult mortality. So I take questions or arguments. I take it, I am taking it that I am hearing some skepticism on these simulation claims that worrying about the last few hundred generations is not something I have to worry about. Am I getting the right message?
It's just like you said, where you kind of match 1 over s to the time scale. I think if your s's are not so much bigger than 1 over the time, this time scale population growth, I'm really worried. But
Yeah, I guess it seems like the prior on when mutations arise is strongly influenced by population size. So the very large population size recently seems to be putting a lot of mutations in.
But and a lot of people. So these are, remember, rates per person.
Yeah.
What Tennyson and company were most concerned with is the whole ensemble. You get hundreds of thousands, and most of them come recent, but you've also got a lot more people to divide them by.
Well, there are a couple of things. I mean, first on the thing of exponentially growing population size, you tend to have long tips on the, genealogies, which will allow for more accumulation of mutations. And a different thing I worry about is sort of population structure. There, things like Ashkenazi Jews, and sort of populations with small, effective population sizes that harbor deleterious mutations.
In this world, I am a consumer. I know that these are problems that don't have the expertise to solve, and I am eager that the population genetics world tells me what to be putting in. I agree, and when I lie in bed and think about these things, population structure worries me a lot, because for a demographer, effective population size is a very strange thing, and happily, we're talking about spatial structuring, and the breeding pools are small things, and then you ask, well, do they really spread out all across Europe in 50 generations? There's a study by Rodi Chang and Olson, which claimed that the most recent common ancestor of all of us lived at about the time Julius Caesar was murdered. He actually tracked flows across Alaska and so forth.
No, is that somebody
So
that gives his DNA to everybody on the planet?
no, because the DNA goes up linearly. This is genealogical ancestry. I'm basically agreeing with you.
Oh, okay, but
No, this is genealogical ancestry.
2 to the n.
Yeah.
This is a 2 to the n, and
We're all related.
genetic
to
ancestry goes up like n or 2n.
Yeah.
And so, no, it's not at all. But in terms of the anthropology of whether small flows over hundreds of generations mix people up, some part of me thinks at random mating very odd here, and some part of me thinks, well, hundreds of generations, very long time, and I don't know where to go between it. But yes, I know I won't solve this, but it is something I worry about at night.
Yeah, so I think the things that I would be concerned about most would be epistates, dominance, effects. I don't. Maybe those don't matter in some way, but for people, it's different things. So, and just also, I wasn't clear. Like, if you put in,
Well, remember, these things are pretty rare, although they're not, I've just discovered they're not as rare as I thought. I thought they were going to be quite rare, and in fact,
was it dominance or mutation?
no, and if they're rare enough, you don't get homozygotes.
Right.
So, but they're not as rare as I was expecting them to be when I started understanding what was psych frequency spectra. So the issue you're raising comes back at me. If these things were very rare, then I wouldn't worry about it. But keep going.
And just wait. So is it? I just understand. How did you get like the peak of it coming at 10 to the minus 3? The kind of main effect coming from alleles of size 10 to the minus 3. Just intuitive the word.
Well, I just looked at what people were simulating in the genetic literature. So.
It's just, I don't know.
There was some calculation you ended up with 1 over 1,500 by doing something. Right?
So, what's the world of understanding is most mutation. A lot of these single SNVs are totally neutral. A lot of them are roughly neutral. People plug in gamma distributions, and the cutoffs, when you've got an effective population size of 10 to the 4, then somehow what neutral means is 10 to the 4-ish, and what strongly selected means is 10 to the 2-ish. I'm not.
Yeah, but I think you're asking about the calculation you had for why the MA alleles, why the important regime for the MA alleles is 10 to the minus.
I'm getting this out of my Gesset flow divided by my Gesset stock.
Okay. So to kind of balance the influx and stuff.
That's it. Well, and I have a closed form adjustment for epistasis. That's what makes this. That's what we have to offer to a wider community. There's actually a closed form adjustment to Haldane's principle. So it's not just we've got this fitness and then there's some fudge factor or other. We've got this fitness and then we can calculate the fudge factor. So that's where I came up with the 1,500.
Wait, but then you calculate kind of influx of mutations times fixation probability. With this kind of curve, it comes out consistent.
None of these are. None of these are going. Are once they fix,
Oh, sorry,
they're
sorry,
gone.
probability of reaching
These are all in balance down at the lower
probability of
end.
reaching some frequency.
And remember, once they fix, the feedback adjusts for them anyway. So the fixes are gone in this picture.
Okay.
Graham has another comment.
So it's now known that the male input of mutations is age dependent, so that there's a nice sort of almost linear increase in mutation rates with the age of the
Father, yes.
How does that get incorporated in this, or how should I think about that?
That doesn't get incorporated at all, but as you say, that's the big recent study, and I know Greg Cochran and Henry Harpending and others, a lot of people are now trying to see how that matters. And again, I'm on the consuming end of that research. I want to find out how it matters. But that study, where they're seeing it, that's up in the 70. They're talking about a pattern of mutation rates with paternal age in an ensemble of 70 things per person in these families that were with what is it, schizophrenia or some bad condition they're looking at where they've got pedigrees. And so that's out of 70. Whether that's so important for my 0.2 out of 70. Oh, I don't know. I'm waiting to see. But there's some interesting stories, I understand, being put together, predicated on that. And of course, because of the different ages of different ranges of male fertility, it might well come into that calculation I'm doing. But I'm waiting for more literature I can draw on.
Do we have time for?
David's the
Do we have time for one more question?
gatekeeper.
Yep.
So David, yesterday you were making an assumption about convexity of the.
Which turned out to be concavity.
Right, yeah.
Misprint for concavity, yes.
Can you remind us? So is that an assumption here, and can you remind us what that means?
Well, here it just follows from, so there's a general model which can have any cost function in it, which the mathematics works for. Then we put in the demographic cost functions that I've been showing, in which there are age-specific profiles for effects of alleles. The concavity is automatic in that case, because what it's saying,
Die once.
die once, you can't die again. That's the concavity.
Okay, but is this, so can you have combinations of alleles that are static and that they're only bad if they're together?
Oh, yeah, so none of that is in here. So this is one piece of epistasis. I'm trying to spell out on one piece. There are all kinds of other pieces, which have been talked about here, where you can have critical points in some gene network that are knocked out. And so none of that's in here.
Do you have a sense if that changes things?
Well, it would certainly change outcomes. The question is how dominant is that for these kinds of things that operate over very long time periods. Knocking out something in a gene network, as we heard, kind of a big effect. That's what gives the fat tails, and that pushes us out where these things have to be understood in recent environments.
You could have two mutations in the regulatory region that is a relatively small effect that still takes a
Yeah, you could. All that, none of that's in here. All that might affect the more refined picture that we'd like to have in the future.
Okay, well, thank you very much.